To go into our first session. This session is entitled Psychiatric Disorders and Precision Neuroscience. I'll be moderating the first session. These are the speakers, and our very first speaker is Dr. Susan Amara. Susan is now directing the intramural program at the National Institute of Mental Health. She's a member of the National Academies of Science. She was a Howard Hughes medical investigator. Prior to joining NIMH, she was at the University of Pittsburgh, where she was chair and distinguished professor of neurobiology. And she has a very illustrious career as a researcher, working in the neurobiology of mental illness. And she has recently taken on major leadership roles at NIMH in addition to continuing her research. She served as the president of the Society for Neuroscience previously. And this is not her first visit to the VTC in Roanoke, so we're very delighted to have her back. Please join me in welcoming Dr. Susan Amara. Susan. You got her phone? Okay. Is she wearing the headphone? Okay. Would you rather stand and talk into that? Whatever. This way you can walk around.
It's faster.
Okay.
It's faster to just start talking here.
And here's your advance.
Great.
And get laser pointer.
Great.
Okay.
Yeah, I'm going to just advance it on my own. But at any rate, it's a pleasure to be here. I can see familiar faces. I have to say I've had a number of wonderful experiences in my own laboratory. At least 3 of the institutions you had on your list I've had fellows, visiting scientists from, and so it's wonderful to be here. And the NIMH actually has a graduate program in partnership with the Karolinska Institute. So the National Institute of Mental Health has this joint program. I wasn't sure how I was going to speak to precision neuroscience. And so what I thought I would do, especially in 20 minutes, I thought I wanted to talk about science. And what I decided to do was to really tell you a story about how I think the sort of elegant and intricate workings of signaling pathways really allow one to target things in very unique ways. So I'm going to be talking about drug action, and I'm going to tell you a bit about mechanism. And I think, as hopefully you'll come away with the idea that, in fact, targeting very selective and specific pathways and knowing and understanding how they work within the cell really allows you to think in a much broader way about how to develop drugs to treat mental illness. The work I'm going to talk about today was accomplished by this group of folks, an MD, PhD, David Wheeler in the lab, Suzanne Underhill, a research fellow in my group, Patrick Houlihan, a postbac fellow in my group, Conrad Hong, a former postdoctoral fellow in my group, and in collaboration with a group at Oregon Health and Science University, headed by Susan Ingram. And I'll just start you off on the topic of amphetamine action. And this is how I learned how amphetamines work. First of all, I should preface by saying that amphetamines are used on a daily basis to treat kids, so to treat ADHD. And in the US, approximately 6.4 million children ages 4 to 17 have been diagnosed or received a diagnosis at some point in their lives. And most of those receive either methylphenidate or amphetamine as their treatment. And it seems to me we ought to understand how amphetamines work. So this is how I learned how amphetamines work. They're substrates for the dopamine transporter. They inhibit dopamine reuptake, and they potentiate the actions of dopamine in that way. But distinct from a lot of other psychostimulants, amphetamines enter the cell, and they also have actions within the cell. The actions that have generally been described involve interactions with a vesicular monoamine transporter on vesicles, provoking the release and essentially facilitating efflux of dopamine from vesicles, and then ultimately leading to a reversal of the dopamine transporter and a potentiation, further potentiation of dopamine. But if you actually work with these drugs for a while and you study their mechanism, there are a lot of things that don't quite fit with this model. There are a lot of phenomena that occur. And so there have been a number of revisions to the way we think of amphetamine action over the past few years, and this has been really the contributions of many groups. One is that efflux via the transporter is actually regulated. It's regulated by phosphorylation at the C-terminus of the transporter, shown by Jonathan Javich and colleagues. It's regulated by CAM kinase 2, through an interaction on the C-terminus of the carrier. It's also regulated by PIP2 within the membrane. And recently, members of my group have shown that G-beta gamma subunits actually regulate the efflux properties of the transporter, and actually release of beta gamma upon receptor activation leads to an increase in efflux. And so it seems to be a little more complicated. But there was another observation that we found particularly interesting, and that was that endocytosis of the dopamine transporter seems to be activated by several DAT substrates, and these include intracellular amphetamine, methamphetamine, and dopamine, amongst a number of other compounds that regulate this. So the phenomena is illustrated in this slide, and it'll be a movie, and you'll see what happens to the cell when you apply amphetamine. This is just a hex cell, nothing particularly special about it. It expresses the dopamine transporter, and we're looking at events that involve internalization events of a fluorescently tagged DAT from the cell surface using turf microscopy. I'm not going to go into the details. So wait for the arrow, and you'll see that upon application of amphetamine, you'll see an increase in the number of puncta that represent essentially internalization events that involve and include the dopamine transporter. And actually, this is in SK and SH cells. So we went on to think about what the mechanism was. And it turns out there are a number of mechanisms for endocytosis. Most people are familiar with the clathrin and dynamin-dependent mode. We checked that, and this endocytosis didn't seem to be, in studies I won't have time to talk about, clathrin and dynamin dependent. Didn't seem to be caveolin and flotilin dependent. Oops. However, when we looked, it's not moving forward. Let me see if I click it here. Okay, great. What we found was it was a dynamin-dependent process, but it didn't involve clathrin. And we began to explore the possibility that it was mediated by the small GTPase row A. To do this, we used a botulinum toxin that inhibits row A-dependent internalization. And we were able to show that, in fact, the process of internalization could be very nicely blocked by C3 exotoxin, which activates row GTPases, really demonstrating that this is likely to be a row A-dependent event. This isn't all this. This really shows, and we also used a dominant negative row A that, to show that it was row A dependent as well. I'll go on to the next. We were able to show these same events occur in neurons, and this is using a surface label dopamine transporter and washing it traffic into the cell. And again, amphetamine produces this decrease in cell surface dopamine transporter. But the curious feature to this process was that it could be blocked by another psychostimulant, cocaine. Cocaine essentially binds to the transporter and prevents internalization, and thus prevents the movement of substrates into the cell. And so this gave us a clue that in fact, it required amphetamine within the cell to activate these processes. So we also showed that amphetamine activates both small GTPases, Rho and RAC, in acute brain slices within the cell. And again, the activation of Rho can be blocked by cocaine, suggesting that amphetamine needs to enter the cell to have these effects. So we were also able to show that the events we observed within the brain when we looked for activation of Rho A really were selective for dopamine neurons. And this is using a GST affinity probe to identify areas of the brain where Rho was activated. And if you'll notice, as compared to control, the main area that we see Rho activation really corresponds to Th-positive cells within the midbrain. And it suggests that, in fact, the Rho activation we saw in brain slices was indeed occurring within dopamine neurons. So it's just not responding all the time. So the question arises, how does amphetamine activate these intracellular signaling pathways? And so I've shown you that amphetamine entering the cell through the dopamine transporter activates Rho A. I won't have time to show you, but another phenomena that we observed was that cyclic AMP goes up when you apply amphetamine to actually neurons and other cell types. And that this, we knew already that this particular pathway, activation of it, could inactivate Rho. And I won't have time to tell you the studies, but the actual window of internalization of the dopamine transporter is determined by the timing of activation of PKA versus activation of Rho A. So what could be doing this? And we went on to, we had worked some years ago on a group of GPCRs called traceamine-associated receptors. And these are a GPCR family member that is activated by a variety of trace amines, biogenic amine metabolites, and a variety of drugs as well. It has a fairly broad range of ligands which activate it. They include paratyramine, beta-phenylethylamine, which has been proposed as an endogenous amphetamine. And they were known to be coupled to increases in cyclic AMP. When you express them at high levels, they reach the surface, and you can study them by looking at increases in cyclic AMP that are activated. However, the curious thing about this receptor subtype is that it is predominantly intracellular in most of the neuronal populations it's observed. And so this became a really good candidate for a receptor that might act and be activated within the cell that could mediate some of the effects on trafficking that we observed. And so we began to explore that. And the next slide simply shows that just in a simple cell line, if we delete the endogenous TAR1 receptor in HEK cells, we can prevent the internalization of the dopamine transporter, and this just illustrates that. And if we reintroduce TAR receptors back in, we regain sensitivity. If we go on to look at TAR1 knockout mice, you can see that very nicely the TAR1 genotype confers essentially the ability, if it's in the wild type, you see very nice downregulation of the transporter, but if you look in the homozygote knockout, you lose that capacity, and the heterozygote seems to be intermediate. If you look at Rho activation, you see very much the same thing, where you see nice Rho activation in the wild type, but really no, or little or no Rho activation in response to amphetamine in the TAR1 knockout mice. So we went on to ask questions about what the coupling was of this receptor that was signaling within the cell. And what we did was we took advantage, Heidi Ham's group had made a series of mini genes that allowed you to probe, essentially generate a C-terminal fragment within the cell that would allow you to interfere with the coupling of different G alpha subunits. And so essentially you could use these mini genes. We went on and we actually just made cell permeable peptides that correspond to these to really piece together what the G alpha subunit that was the effector that was activated by TAR1. And so we showed the experiment worked rather nicely in that what we were able to observe was that a single G alpha subunit, if you interfered with its function using these peptides, you could actually block the effect of using one that corresponded to G alpha 13. G alpha 13 turns out to be the effector for Rho signaling. And so it really suggests that activation of TAR receptors within the cell couples through G alpha 13, and that's linked to Rho activation. And we could show similar things occur in neuronal cultures as well. So we also looked to see what else was going on within dopamine neurons. And in particular, over the last few years, people have shown very elegantly in many different ways that dopamine neurons are glutamatergic. And they express other peptides, but they also express vesicular glutamate transporters, and they express plasma membrane glutamate transporter, EAT3. And what when we looked at what happened to EAT3 within these cells in response to amphetamine, we saw that like the dopamine transporter, amphetamine produces a decrease in cell surface, the EAT3, and thus reduces the ability of those cells to clear glutamate. And like the dopamine transporter, EAT3 internalization is dependent on activation of Rho-A and dynamin, the small GTPase that's linked to the internalization process I was describing. So this suggests that glutamate transporters also were being taken off the cell. So what happens in slices to glutamatergic activity? And in this case, Susan Ingram was able to show that you can actually, using amphetamine, you can potentiate glutamatergic currents. Now these are currents where you're recording from the dopamine neuron itself. So these are cells that are releasing glutamate, they're cells releasing glutamate onto the neuron. You record from the dopamine neuron and you record glutamatergic currents. And what you can see here is that you get a potentiation of both NMDA and AMPA currents, which is sort of consistent with the idea that you're removing a transporter from the surface to have this potentiating action, because it's really agnostic to the receptor subtype. And so we went on to do other experiments to show that this was due to removal of the transporter in that particular cell, and confirmed, in fact, that the effects of amphetamine have effects on glutamatergic signaling. We went on to identify a C-terminal part of the carrier, the EAT3 carrier, that if we introduced a cell-permeable peptide corresponding to it, we could, this particular region confers sensitivity to amphetamine. So we could then generate a cell-permeable peptide that corresponds to this region and includes this critical sequence, VNGGF, and we could essentially use that to independently alter the glutamatergic signaling within the cells, as opposed to just what was affected by the changes in the dopamine transporter in that cell. And this simply shows that the EAT3 peptide blocks the effects on currents and it also alters the, prevents the downregulation of EAT3 from the cell surface. But it has no effect on the dopamine transporter. And so, but no effect on the dopamine transporter. So then we began to explore further and show that in EAT3 knockout animals, amphetamine doesn't potentiate. In this case, it's the NMDA EPSCs in EAT3 knockout mice, suggesting that, that this potentiation is in fact due to internalization of EAT3. And in the EAT3 knockout, there's a curious effect of acute amphetamine, and that is that if you look at amphetamine in an EAT3 knockout animal, you see that amphetamine has very little stimulatory effect on locomotor activity, suggesting that some of the acute effects of amphetamine are actually due to its changes and the way it regulates glutamatergic signaling. And so I have one last data slide before I kind of summarize, and that is that we then went on to look, and I'll pull this in at the end, we then went on to look at what happens with methamphetamine. So if you apply, this is using a very nice FRET sensor generated by Mark Rizzo, and this detects activated Rho. If you apply amphetamine to a cell, this is a neuron expressing the dopamine transporter, you see this slow rise in what would be activated Rho. But if you don't have the dopamine transporter for amphetamine to enter via, you see nothing. So it essentially blocks the effects. But if you apply methamphetamine, you see an early phase, even in cells which lack the dopamine transporter. And what this tells you is that meth is likely entering the cell via a different route and doesn't require the DAT to get in. Meth also has this slow phase of where it activates Rho, and that is DAT dependent. So it suggests that meth has the ability to regulate glutamatergic signaling much more broadly in the brain through its ability to alter the trafficking of the glutamate transporter. So now I'm just going to come to my summary. And so what I really wanted, well, that isn't my summary yet, okay. So I've tried to kind of illustrate a number of pathways within the cell that are involved in the actions of amphetamine. The first is the cell surface receptors, and this is something I didn't show you because I didn't describe the cyclic AMP pathway in detail. But it turns out what you can do to modulate amphetamine action is activate cell surface receptors that couple through cyclic AMP that either activate or inhibit and alter pKa to prevent, essentially, if you activate it, cyclic AMP and pKa, you can actually block the acute effects of amphetamine, not only on Rho A, but on behavior. So the second area that I've tried to allude to is that amphetamine also enters the cell and activates a trace amine receptor. And we've been using FRET sensors to identify the compartment in which it signals. It doesn't signal from the surface, and it does signal from an intracellular membrane compartment. And that leads to the activation of Rho and changes in dopamine transporter trafficking. So the third place where you can see that is, again, potentially targetable in terms of drugs to add greater selectivity is the idea that we can interfere with the internalization of EAT3 independently from the internalization of the dopamine transporter, and in that way modulate the actions of amphetamine in yet another way. And finally, the actions of amphetamine are determined by whether the drugs are selective for the transporter or whether they have some other mode for permeation as we observed for methamphetamine. And so taken together, exploring this sort of more detailed analysis of the intracellular signaling pathway, I think gives us opportunities to target not only cell surface receptors, but intracellular receptors, as well as the internalization mechanisms themselves, which we're currently working out. And that, I think I can take questions. I can summarize. The important points are that amphetamine acts intracellularly to activate Rho A and RAC and trigger internalization of the DAT. PKA activation inactivates Rho and inhibits the acute behavioral effects of amphetamine. That's data I didn't show. This is real. And then activation of TAR receptors within the cell is an essential element. And a number of drug companies are currently exploring TAR receptors as a drug target in a variety of psychiatric diseases. Amphetamine also it triggers endocytosis of a glutamate transporter and modulates excitatory signaling. And then the possibility exists that compounds that permeate the cell but yet can activate the trace amine receptor can have effects on Rho signaling and other intracellular signaling pathways that are even independent of the dopamine transporter. So that's it.